DNA extraction. I'm going to extract the DNA from this, a rat's tail, and we're going to extract the DNA from the cells in which it normally resides. I'll use an enzyme to digest the proteins and a buffer containing a detergent to break down the fatty membranes. To remove the DNA from animal tissues, I'll start by cutting up the rat's tail. This Eppendorf tube contains the cut up rat's tail. To this, I'll add the enzyme and a buffer. The buffer again is to break down the fatty membranes. Then this will be incubated at high temperature, and the high temperature is to increase the activity of the enzyme. After an overnight incubation at high temperature, which was to digest the proteins and break down the membranes, we can then purify the DNA. This is a centrifuge which will spin the sample up to 13,000 RPM, separating the cell fractions. Now the DNA has been purified, we can amplify specific regions using the PCR machine. This stands for polymerase chain reaction. Through a series of hot and cold cycles, this separates the DNA strands and copies them, giving us a lot more product to work with. Once the amplification cycles are finished, we drag the DNA through a gel using an electric current. The DNA can then be visualised using a stain in UV light.